Greetings, dear learners. In the last class, we learned that for the purpose of chemical analysis, poisons are grouped according to the methods used for isolation of substances from the matrices. Under that, for the purpose of chemical analysis, poisons are grouped into the noxious gases, volatile poisons that includes organic as well as inorganic, then non-volatile poisons that both organic and inorganic, then certain plant poisons and miscellaneous group of poisons. Then we also understood the need of extraction of the poison from the large biological matrices before applying the analytical techniques. Under that, we learned certain important and commonly exercised extraction methods that included solvent extraction, distillation, fractional distillation, distillation under vacuum, sweep co-distillation, microdiffusion, dialysis, sublimation, digestion or chemical treatment, microwave digestion, absorption chromatography. We also knew the classical and modern methods of the extraction of different poisons. Under that, we knew for the gases, microdiffusion, adsorption, desorption, modern techniques, gas analyzer, gas chromatography. Similarly, for the volatile inorganic poisons, volatile organic poisons, then non-volatile inorganic poisons, non-volatile organic poisons, then for anions. Then, under the modern method of extraction, we knew that the modern methods includes various methods in chromatography as well as the special extraction methodologies of the present generation like ion pair formation, solid phase extraction, solid phase micro extraction, Micellar extraction, then certain special type of digestions like isolation of toxicants through the chemical processing that includes the microwave digestion, digestion by the plasma source for the inorganic sample are also an emerging technique. The other methods including hyphenated techniques like HPLC-GC, SPC-GC, GCMS are under the processing. So we will be now learning some of them in detail. Starting with the head space technique. The head space method is especially suitable for the very fast separation of volatile components, alcohol, acetone, aldehyde, in a complex biological matrices, especially blood. You can see in the picture when the sample is in the tube or a jar, then this empty space is called the head space. So it is the principle behind this method is that the whatever in the volatile substance, what is present in the solution get diffuses into the air and there is a equilibrium established between the air and that liquid. So by just injecting sensor from this tip on taking the air sample, we can analyze the sample and we can detect the what exactly amount this whole system contains. So this is a way of deciding for the head space technique. So under this method, the advantage is that the risk of contamination of non-volatile components may be eliminated for online analysis of toxicants by the gas chromatography. Then it is analyzed for the gas chromatography and then further analysis by the mass spectrophotometry may be done. The principle underlying the head space analysis is that in a sealed vial at a constant temperature, an equilibrium is established between the volatile components of a liquid sample in the vial and the gas phase above it, the head space. After allowing the time for equilibrium, that is normally 15 minutes or so for 50 or more samples in a single run, a portion of the head may be withdrawn one by one from the vials using a gas-tight syringe and injected to GC online for further analysis of the separated compounds. So in this way, this head space technique is used. Then another technique is the solid phase extraction method. In this method, siliceous material with a relatively close size distribution and various silica bonded phases like noctadecyl, endocyl, N-hexyl, ethyl, methyl, cyanopropyl, aminopropyl have been employed as adsorbent with much greater efficiency of separation. So this on the solid phase, that is the stationary phase, some adsorbent of good quality is added that helps in the separation of the analyte. There lies however, a recently developed type of the solid phase extraction cartridge in which very small particles of the adsorbent are enriched in with the PTFE microfibrils, having the similar efficiency as in packed column but require less pressure drop. The sample is applied to the SPE cartridge either from a syringe or sampling manifold by applied pressure or suction at the lower end. The components are subsequently recovered by solvent flashing. The method is attractive because of the small amount of sample and material necessary and also much greater speed of procedures compared to the classical adsorption method. So you can see in the picture here this jar is containing the biomatrix of our interest that contains the interfering components as well as the target molecule. So here the interfering components are then administered, this solution is administered by the adsorbent that is of interest that combines with the analytes of interest, and that will form a complex with it, and then the other particles, the interfering components will be excreted out, whereas the analyte of interest will remain inside attached with the adsorbent, and when washing will be done, the other interfering component will be excreted out, whereas for the eluting, that is a step involved where along with the adsorbent, analyte of interest will be taken out and it will be analyzed further. So this is the idea behind the solid phase extraction. Then another modified technique is the solid phase micro extraction. In the solid phase micro extraction, it is an extraction technique for organic compounds in aqueous samples in which the analytes are adsorbed directly from the sample onto the fused silica fibers. So here this jar is already having the fused silica fiber, there it will attach, and we will remove this silica fiber and they will be subjected to the further analysis, GC and the MS. So the other way around, in the solid phase micro extraction, they were getting extracted outside in jar, whereas it is the fiber, they get attached to the fiber and we will test it further for using the GC and MS. So here organic compound in aqueous samples in which analytes are adsorbed directly from the sample onto a fused silica fiber that is coated with an appropriate stationary phase. When the fiber is inverted in the sample, the analyte portion from the sample matrix enters the stationary phase until the equilibrium is reached. The fiber is then inserted into the injection part of the gas chromatograph, where it is heated and the analytes rapidly thermally destroyed into the capillary GC column for the analysis. So in this way, solid phase micro extraction is done. Then supercritical fluid extraction. Here the gases above their critical pressure and temperature are in a supercritical states. Intermediate between that are the gas and the liquid. Supercritical fluids have a strong extraction properties because of the solubility of components in the fluid is close to that of a true solvent, and much lower viscosity allows it to percolate through the packed bed of samples. Thus, not only there is an efficient contact between the extracting fluid and the sample, but the fluid is easily removed when it is released from its supercritical state. So, in this way, with the help of the supercritical fluid, we can extract the analyte of the interest. Then another technique is the Micellar formation. It is same as the soap technique of soap, where the fat is removed with the help of the soap particle. We already know how the micelle is formed, that has the polar and non-polar ends. So in the micelle, they gets into two ends. One is the polar and the non-polar end. Polar end is hydrophilic in nature, that gets attracted to the water, whereas the non-polar end is hydrophobic in nature, that is opposite to the water, and that will surround the wild drop or the metabolites, or the biometrics of our interest, that has to be fat contents and everything that has to be removed. So in this way they will form a micelle around the fat molecules, and they will get excreted out by the washing it. So in this similar way, the sodium and the cationic side is the polar end, that is on outside, and this will form as complex with the vial or grease, and this is the micelle part. So micellar extraction is a special type of extraction procedure that appears to be unique in the separation of drugs, plant poisons and the pesticides in the biological matrices like viscera. In the extraction of active constituent, micellar involvement of the surfactant of different classes is employed. Surfactant in the solution also act as the interface of the two-phase system of the vial and the water, or organic solvent and the water, resulting in the solubilization of the phase into one another. An emulsion or the microemulsion is formed by the process. Biological matrices, that is a viscera, in the Indian perspective contains the fats, degraded proteins and the coloring matters. The resulting extraction of the active constituent becomes difficult. In the solvent extraction process, if the surfactant is added to the extractant, deproteinization and the also solubilization with the formation of the emulsion occurs due to the Micellar interaction. The emulsion thus formed is due to solubilization of fat in the biological matrices in added solvent containing the traces of water, with simultaneous formation of emulsion occurs due to Micellar interactions. The emulsion thus formed due to the solubilization is destabilized on increasing the concentration of the surfactants in the system. As a result, fats are separated as semi-solid material due to lowering of the zeta potential between the electrical double layers and the colloidal system. As proteins and fats are separated out, the supernatant liquid containing the active constituent may be extracted for poisons by the organic solvents. So in this way, the remaining filtrate is containing the analyte of interest, and that may be checked through the GCMS or other techniques. Then another technique for the extraction is the microwave accelerated reaction system, where the method of extraction is used for isolating pesticides in biological materials, especially in the kidney and the liver. In this process, the sample is subjected to rapid heating with the organic solvents by the microwave at an elevated pressure, resulting in isolation of the active constituents. The biological material of interest is placed inside the microwave. This type of the microwave that we are using in the home is used in the labs as well. And so this biological material is placed inside the microwave transparent vessels with a polar solvent ionic solution, usually an acid, and is subjected to rapid heating by the microwave in the microwave accelerator system, that is also known as digester. The method finds application in digestion of the biological materials for isolation of some toxic metals like copper, arsenic, lead, and determination of the atomic absorption spectrophotometry and ICP. The method finds application in digestion of biological material for isolation of some toxic metals like copper, arsenic, lead, and determination using atomic absorption spectrophotometry and other methods. Then another technique is the universal trace residue extraction. Here it is a system that has been developed for the recovery of pesticides and organic residue from the wide range of samples including the biological materials. It is based on the principle of sweep co-distillation that relies on the preferential volatilization of the pesticides or other organic chemicals from the biological materials, lipids, plant extract, and the subsequent isolation of volatiles on the cold traps of solid adsorbents. It is a purge and trap technique involving dispersion of the sample in a thin film on deactivated glass beds at the elevated temperature. The extractor system is specifically designed to recover volatile thermally stable organochlorines and organophosphorus from the lipids, meat, butter, viscera and others. At present, the distillation tube doesn't contain glass beads or glass wools as it renders less recovery. Important to know here is the fluorosil, that is activated magnesium silicate in conjunction with the sodium sulfate, has been found satisfactory for trapping many different classes of volatile organic compounds like alumina, silicazole and Tinex are the material that have been potential for use as a trapping media with advantage over fluorosil in the specific applications. The method is expected to fail for the thermally labile pesticides. The consumption of solvent is minimum. The method requires the optimization of the analytical conditions before the application to the biological samples, especially in the forensic cases. However, it is a two-in-one process where the extraction cum cleanup is a very short time with a very economical use of organic solvents, unlike solvent extraction methods. Another method is the accelerated solvent extraction methods. Here the name of the method signifies that multiple sample handling in a very short time by a very updated extraction system, which also works in the same manner as in the case of the universal trace residue extractor, that is the sweep co-distillation. Sweep co-distillation method we already saw in the last class. The same principle is behind the universal trace residue extractor and the accelerated solvent extractor. In this method, a commonly used solvent is pumped into an extraction cell containing the sample, which is then brought to an elevated temperatures and the pressure. Minutes later, the extract is transferred from the heated cell to a standard collection vial for the cleanup analysis. The entire extraction process is fully automated and performed in a minutes for the fast and easy extraction of the multiple sample with a very minimum solvent consumption. So this is the basically extraction cell where the high temperature and pressure used for the extraction, and after the extraction the sample is collected in the collection vial, which may be subjected to further analysis. So this is a less time consuming and minimal solvent consuming procedure. Size exclusion chromatography is another technique. The technique can be used to the advantage as a preparation technique for the prior fractionation of vials, fats, environmental samples into the discrete molecular weight fractions, and as a way of removing very high molecular weight material from the complex samples. It helps to generate high quality chromatographic result by using organic solvents at a high temperature and pressure to quickly and efficiently remove unwanted matrix component from the sample. The extraction cell is filled with a solid and semi-solid sample that you want to analyze. This sample matrix can be complex such as soil, food or the plant material. The cell is heated at a constant pressure of around 0.3 Pascal up to a maximum of 200 degrees Celsius. The elevated temperature increases the extraction efficiency of the analyte of interest, while the elevated pressure keeps the solvent in the liquid state as the temperature rises above its boiling point. The sample is kept at constant condition for a while to establish an equilibrium. During this time, the analytes are extracted from the samples into the solvent. The extract is then transferred to the sample tube for further analysis. This is the concept behind size exclusion chromatography. Then the ion pair extraction. Here actually this, as we see here in the picture, this sodium chloride is a compound, whereas the sodium in hydrated form surrounded by the water and the chloride in aqueous chloride surrounded by water both attract to each other, forming the ion pair. They don't form a sodium chloride, but they are just getting attracted. This is called the ion pair formation. So this basic method idea is used as for the extraction method. This method is applicable for the extraction of highly water soluble organic compounds. These compounds cannot be extracted from the samples by direct solvent extraction. The difficulty has been overcome by forming ion pair with a suitable reagent like quaternary ammonium salt as the ion pairing agent. The ion pair formation is the attraction between the positive and negative charge. This is not a reaction and it is only an attraction. Once this pair is formed, it will act exactly like an organic molecule and not like an ionic compound at all. The extraction may be carried out thereafter by the direct solvent extraction, preferably in the presence of a buffer. The method is also applicable for the extraction of the drugs in form of the quaternary salt in urine or blood. The biological matrices are deproteinized and treated with the dye in the presence of buffer to form a drug dye complex, which is extracted by the organic solvent. The dye is destroyed by the action of acid or alkali and the drug is isolated for the analysis. In this way, the ion pair extraction method is applied. Then another technique is the hyphenated techniques, where the more than one process are combined and a more better extraction is done, like HPLC-GC, SFE-GC. So the hyphenated techniques like HPLC-GC and the SFE-GC allows the complex samples to be pre-fractionated rapidly according to the molecular weight or chemical classification on a suitable HPLC. We will be detailing on the methods of the HPLC gas chromatography in the next class. This HPLC pre-column or SFC and appropriate fractions then separated by the line capillary gas chromatography. We will be also knowing the methods or the steps involved in gas chromatography. Basically, chromatography makes the separation based on the affinity of the solvent with the mobile and the stationary phase of the column or the gas chromatograph. So, based on that, this separation that happens and the extraction procedure is completed. The coupling of two techniques has been made by the bypass valve fitted with a sample loop of appropriate volumes. The selected fractions are passed to the capillary column via a large retention gap, which utilizes the process of concurrent solvent evaporation to retain the fraction in the retention gap, thus avoiding any preliminary contact with the column. Selected fractions are passed to the capillary column via a large retention gap, which utilizes the process of concurrent solvent evaporation to retain the fraction in the retention gap, thus avoiding any preliminary contact with the column that must be adjusted suitably. Then for the extraction of the non-biological matrices, chemical analysis by preparing the solution of samples and the systemic group analysis is sufficient. For the biological matrices, the extraction of metals in the biological matrices may be carried out by the following the methods of the dry acing methods, wet digestion method, acid digestion method, Fresenius and Babo method, selective chemical treatment. Actually, these are already the extraction, most of the extraction methods we already discussed. But for some particular type of the agents, particular type of the matrices and the particular type of the substance, different specific methods are applied or in the process of the development. Like in case of the metals, dry acing method, wet digestion, acid digestion method. So this is the modification of the digestion method only. Then Fresenius Babo method, selective chemical treatment. The organic matters which constitute the bulk portion are destroyed by the chemical means to get the active constituent metal ions free completely for qualitative and quantitative analysis. Here in these methods, the organic matters which constitute the bulk portion are destroyed by the chemical means to get the active constituent, that is metal ion, free completely for the qualitative and quantitative analysis. Then coming to the brief detail of these methods, in the dry ashing methods, about 10 grams of the matrices is taken in a silica crucible and heated in the Bunsen burner for removing the moisture and partially destroying the organic material. Then the crucible is kept in a muffled furnace. The temperature of furnace is raised to 5500 degrees Celsius, and at this temperature, the incineration of organic matters is performed by keeping the silica crucible for an hour. After incineration is complete, the crucible is taken out. The color of the residue is noted when hot because in the presence of the zinc, the residue assumes yellow color, while in the presence of the copper, the color is the bluish green. So, at this stage, noting the color may itself suggest the type of the metal it has been. Further, it is tested for the metallic poisons such as copper, bismuth, zinc, barium by performing the general group analysis by using the micro methods, chromatographic and instrumental techniques. So, this is the way to perform the dry ashing method, means let the biological matrices to dry ash. Then wet digestion method. In that, about 100 grams of the biological material or the matrices or blood is taken into the large Ketgel flask. This is the Ketgel flask, and about 20 to 40 ml of the concentrated HNO3 nitric acid is added to cover the material, and the flask is generally heated in a small flame when the mass begins to liquefy. The heating is continued until the liquefaction of the material is complete. So, in the dry ashing method, it is totally dry process, whereas in the wet digestion method, the matrices is liquefied with the help of the addition of the acid. At this stage, about 20 to 30 ml of the concentrated sulfuric acid are added, and the flask is heated strongly over a wire gauze, and the concentrated nitric acid is added in the drops. That is, by using the dropping tunnel to content of the flask at a rate of about 10 drops per minute, so that the atmosphere in the flask must at no times be free from the brown fumes. Heating is continued until all the organic matter is destroyed and the liquid becomes clear and colorless or straw colored. At this stage, a precipitate may be formed which contains the insoluble salts of the lead, bismuth, tin, barium or other metallic compounds. The precipitate is filtered off and subjected to systemic group analysis and the quantitative determination thereafter as required. So, this is method behind the wet digestion method. Then Fresenius and Babo method, that is especially done for the mercury, as through the wet digestion method mercury compound is not extracted. So, the wet digestion method of destruction of organic matter is not at all suitable for the mercury, which is almost completely lost by the volatilization. So, this Fresenius and Babo method is suitable for the liberation of mercury, although there is a possibility of some loss of the mercury by the vaporization, but this is the most suitable method for the mercury. Then to perform this method, a definite amount of biological material, about 20 to 25 grams of the viscera or 5 to 10 ml of blood is taken into the flask fitted with the reflux condenser. In the case of viscera or the other solid material, sufficient water is added to make a gruel-like consistency. 1/3 of its volume of the chemically pure hydrochloric acid and the few grams of solid potassium chloride is added. The contents are mixed by shaking. The mixture is heated over a wire gauze on a burner flame or on a boiling water bath. A small amount of potassium chloride solution is added time to time and the flask is shaken. Chlorine gas evolves. The heating is continued until the contents of the flask become a uniform straw-colored liquid free from the organic matter except some fatty substances in suspension which cannot be oxidized. If heating for an hour after the last addition of the potassium chloride produces no darkening of the mixture, the oxidation of the organic matter may be taken as completed. It takes 4 to 6 hours to attain the stage. It is filtered and washed with the water. The filtrate and washings are collected for further analysis. In this way, this Fresenius and Babo method is done for the mercury compounds. Then selective chemical treatment methods. Here few toxic metals like arsenic and antimony in their specific oxidation states may be subjected to reduction process by the nascent hydrogen by the reaction between zinc and disulfuric acid that produces the nascent hydrogen. For the isolation of metals in the matrices like burnt bones, nails, hairs, and non-biological matrices like food preparations, drinks, tea, coffee in form of their volatile hydrides. The process may be carried out by the Gudez and Mars-Berzelius method. This is actually two-in-one method for isolation and online detection and determination. So here a chemical is added that reduces the biological matrices and help in the extraction. Then extraction of toxic ions in the forensic matrices. The extraction of toxic anions in non-biological matrices requires very minor processing and the cleanup, whereas the inconveniences is usually felt in the biological matrices like the viscera, stomach, gastric content, stomach was, urine and the blood. For the extraction of the ions, the method that is used are the protein precipitation, dialysis, selective chemical treatment, microdiffusion, ion chromatography. We already saw in that table that for this ion is categorized into a separate category. So certain methods that are useful for the ions are this protein precipitation, dialysis, selective chemical treatment, microdiffusion and the ion chromatography. We will be knowing some brief facts about them. In the protein precipitation methods, a slurry of the sample, that is the minced viscera, about 10 to 20 grams of the stomach content or gastric lavage is prepared. The protein in the sample is coagulated by the adding 0.5 to 1 grams of the ammonium sulfate. It is filtered through the 2 centimeter layer of the cotton wool held in the barrel of the syringe. The excess of ammonium sulfate remaining in the filtrate is precipitated by the methanol, and the supernatant liquid is collected and evaporated to a small volume on a water bath. The concentrated liquid is ready for the analysis of the ions. So here it is same as the protein precipitation technique that you know in the eggs that happens that when heating protein precipitates, and this supernatant will contain our interest of analyte. So we have to test on this supernatant part. So this is about the protein precipitation. Then dialysis, that is that same as the passing through the semi-permeable membrane. So for the separation of ions, the about 10 grams of the tissue is cut into the small pieces and placed in the cellophane membrane made into the shape of the bag. The bag is then slowly rotated in a beaker containing the 100 ml of distilled water by means of electrical motor or the mechanical devices. Dialysis occurs rapidly. After an hour, the water in the beaker is replaced by the fresh water and the bag is rotated for another half an hour. The water is then taken out, mixed with the previous fraction and evaporated on the water bath to a small volume. This filtered water content is actually containing the analyte of our interest, and if necessary it may be tested for the toxic anions. So this is method of dialysis. Then selective chemical treatment may be done for the ions. In that, phosphide and sulfide present in the biological matrix is subjected to the treatment with the dilute acid on the hot plate or water bath, and the toxicants that is liberated in the form of the phosphine or hydrogen sulfide are subjected to the chemical analysis. So this is by the chemical treatment of the biological matrices. Then ion chromatography, the preferential exchange of the ions on ion exchange reagent packed in the column of the ion chromatograph renders the separation of ion by using the mobile phases, usually buffers of the diverse pH. We will be learning details of the ion chromatography in the next class when we will be detailing the chromatographic method. Then the micro diffusion. In the micro diffusion, we already talked in the last class where this is the Conway micro diffusion assembly jar, where the central part this testing sample is kept and that will diffuse out in the surrounding, and from there we can analyze the sample. So this is the micro diffusion. This microdiffusion may be employed for the biological matrices like blood, urine, and stomach wash, and also for the non-biological matrices like water, drinks, tea, coffee containing traces of toxic anions that may be the cyanide, phosphide, sulfides by using selective liberating sealing and detection reagent in Conway or microdiffusion assembly. Conway microdiffusion assembly, as you can see in the picture, is that consists of the propylene cell with a clear polystyrene covers, and the cells have the outermost annular sealing wall, an intermediate annular well for the sample, and the liberating agent and the center well for the reagent, which is used to trap the diffusing gas or the vapor. So this intermediate region and the central region that contains the reagent that will trap these diffusing gases and the vapor, and the intermediate well will contain the sample of the interest. So the sealing agent, usually about 2 ml, is introduced into the outermost sealing wall. An approximate amount of about 1 ml of liberating agent, usually same as the sealing agent, like 10% sulfuric acid, or the sealing liberating agent for the carbon monoxide and saturated sodium carbonate solution for methanol, and ethanol is placed on a pool in the one half of the intermediate well, that is in the sample well. Trapping agent, say the PDCl2 in a care of the carbon monoxide turning to black, acidified potassium 24 hours trichromate turning to green in case of methanol or ethanol. The sample is introduced into the other half of the sample well, taking care that it doesn't mix with the liberating agent. The cover is placed and the cell is rotated to affect the airtight seal. The assembly is fitted back and forth to mix the sample and the liberating agent. The cell is placed on the table and water bath if needed for the reaction. The control sample cell is also prepared using the same reagent but without sample, that is 1 ml of water in place of the sample. The color change in the central compartment is noted. The color change in the central compartment will signify the content in the biological matrix. Then comes the Stas-Otto process. It is basically done for the alkaloid compounds. The Stas-Otto method is based on the distribution of alkaloidal base between acids and the aqueous solution and immiscible organic solvent. Powdered drug is mixed with the water and alkali to make a paste. Alkali is added because it is mixed with acid, tannins and other phenolic substance to make it free of the alkaloids. Thereafter, the mixed drug is extracted with the chloroform or other organic solvents using Soxhlet apparatus. This step is carried out because freed alkaloids are dissolved together with other substances soluble in the solvent. Then further re-extract the chloroform layer with dilute sulfuric acid in the separating funnel and the acid layer is collected. This is because alkaloids are converted into the alkaloidal sulfate, which is soluble in water. Then in the acid layer, water is added so that soluble constituents are passed into the aqueous solution. Then the aqueous layer is mixed with ammonia to make the media alkaline. Then evaporate to dryness and precipitate is collected and dried. This step is carried out because ammonia converts alkaloidal sulfate to ammonium sulfate, which is soluble in water. The free alkaloids which are insoluble in water are precipitated, and then further testing on the alkaloids can be done. So you can see here in this chart, the powdered drug containing alkaloidal salt is subjected with the organic solvent, added alkali or the lime that separates the free alkaloids, that is extracted with organic solvent, and that total extract is concentrated and sick with the acid so that the alkaloids get precipitated. Residual organic fraction is get separated and aqueous acid fraction is another separation. Then make it alkaline with ammonia. Free alkaloidal base is prepared and the solvent is removed and the crude extract of the alkaloids is obtained. This is the basically Stas-Otto process. Further modifications and the further advancement in the Stas-Otto process are there, and that is the recent advancement related to the extraction techniques. Then after once the extraction has been done, then also certain interfering agents are there with the solution that need to be removed. So all the extraction technique that were described above, it is likely that extract will contain interfering substances which will create problems in analysis in various ways. The occurrence is particularly so in the extract from the samples of the effluents, soil, sediments and tissues, or the organic matrices which contain fat, oil and other naturally occurring substances. The long established procedure for the removal of these substances has been to pass the extract through the alumina column and then to separate the target compounds into different batches by passing the clean up extract through a silica column. The supply of material by preparative thin layer chromatography is also followed if the active components are identified by preliminary screening. The purification in the same way may also be achieved by the GC or the HPLC. For the volatile toxicants, stripping may also be done by the GC head space method, purge or trap technique. There are different analytical conditions for the purpose which have been specified in various literatures. Recently, the cleanup has been streamlined by the use of commercially available solid phase extraction cartridge or the disc. Different methods that is followed for the cleanup are the cleanup using alumina and the silica column, cleanup using solid phase extraction cartridge, cleanup using preparative TLC method, cleanup using HPLC method. These are the various methods to clean up the extracted material. So with this, we have completed the pre-analytical step, that is extraction and the cleanup of the extracted material. Thank you.